Hello, I'm Tzu-Yi, the speaker of the online introduction video. On behalf of TMU NLP team from the NLP lab of Taipei Medical University in Taipei, Taiwan. As you can see on the screen, our topic is identifying drug-chemical genes interaction in biomedical literature using the BERT-based Ensemble learning approach from the BioCreative 2021 DrugProt track. We all know accurate biomedical entity relation prediction is essential in numerous biomedical research fields, and due to the current global pandemic, it further triggered abundant studies that provide new biomedical research and knowledge. While biomedical literature accumulated at a record-breaking rate, it has become increasingly challenging to efficiently analyze drug-related information described in the scientific literature. Natural language processing method can provide efficient tools to detect crucial information regarding chemical entities and gene entity in a great deal of biomedical articles. As student doctors and data scientists at medical school, we are delighted to participate in this academically renowned BioCreative workshop. We evaluated our method with BioCreative VII Track 1, which is publicly available on DrugProt corpus that contain chemical and genetic entity annotations. Certain sentences that contain chemical and genetic entity relationship were classified into 13 types. The relationship distribution of trained and developed dataset is described as the data figure. Then, we constructed the distance mechanism and LLR-based trigger word weighting counter as the feature engineering method. For our implementation, we applied BERT with biomedical language representation to develop the relation extraction model. Finally, some submission we evaluated the result and adopted 3 or more models prediction through a voting mechanism to reach ensemble learning. In order to find out the proper pre-trained BERT model for the task, we applied hundreds of biomedical representation BERT model with or without feature engineering. We conducted experiments to evaluate the performance under a 5-fold cross-validation scheme and found that PodMed BERT is comparable with BioBERT and BlueBERT top others by a small margin. Finally, to take advantage of different BERT models, the first submission adopted these 3 models' prediction as an ensemble. And for the second submission, we introduced the distance mechanism with PodMed BERT and evaluate the result with and without distance feature. In the third implementation, we joined different batch size setting sorted models. Last but not least, we applied LLR log likelihood ratio to count the term weighting between trigger word and category. Due to the limitation of hardware equipment, we tried to submit 3 large-scale experiments that are the PodMed BERT model with and without distance mechanism and BioBERT model with distance feature. The final performance table is shown above, including the test dataset and large-scale dataset. The result has shown our 4 submission performance range from F1 score under 1% to 20% in terms of aspect of the test set. This performance of interaction identification is far from our validation experiment. We speculate that this is due to some under-detected error in our program, and the performance of the large-scale dataset range from 56.52 to 58.34% F1 score. In conclusion, we found PodMed BERT with distance mechanism achieved the best performance. As we know, we participate in this workshop today is attempt to do better entity relation extraction. In the presence of increasing amount of real-world data, an efficient interaction extraction model using with NLP method is bound to bring out useful information hidden in an enormous amount of text. This work is worth achieving to have machine learn self-predicting on their own to help researchers conduct in more depth. Thank you very much for the privilege of presenting our work. I'm Speaker Tzu-Yi. We are TMU NLP from Taipei Medical University in Taipei, Taiwan.